Hello to my friends joining us via recording. Today in lab, we are going to be working on pages 3 through 6 from your packet, the lesson number 4 packet about the types of epithelial and connective tissue. So we've already split into groups and we've talked about this with some of our classmates. When we talk about epithelial tissue, we can use the name of the type of tissue to give us an idea of what it looks like. So I already have someone that's helped me out down here. When we talk about simple squamous epithelium, we have a note here that this is a single layer of cells. Which of the words in its name, simple or squamous, tells me that it's a single layer of cells? What's the word that means one layer? Yeah, that simple word refers to the number of layers that this type of tissue has. Simple means one layer of cells. Hey, while we're at it, we're talking about simple. Let's bump up to stratified. When I use that word stratified, what does that word mean? What does stratified mean? Yeah, it means that we have more than one layer. So basically the way I like to write it is 2+ layers, at least 2, possibly more. When we start talking about things like the skin, we have lots and lots of layers. Like we can have 50 or more layers of cells. So stratified can mean that there's 2 layers, or stratified can mean there are 50 layers. It just means there's more than one, at least 2, possibly more. This last word that we have right here that talks about our layering patterns is called pseudo-stratified. When we talk about pseudo-stratified, that beginning part, pseudo, means fake. And that word, that part stratified, we just talked about that. Stratified, I can't spell this morning, sorry, means that we have 2+ layers. Yeah, so a couple of us are chiming in the chat. When we talk about this word pseudo-stratified means that basically the way I like to think about it is fake more than one layer. I like to call these guys the fake it till you make it type of tissue, meaning they're pretending that they have more than one layer, but actually it's pseudo, it's fake, it's not real. They don't have more than one layer. They just have one layer. It looks like there's more than one. So pseudo-stratified, only one layer. It's a messy layer, but it's just one layer of cells. The other part of the name of these tissues is things like cuboidal, things like squamous, things like columnar. When I talk about cells that are flat and maybe kind of round in appearance, which of these cells are really skinny, kind of circular, flat? Any guesses which of these words are the, or here, let's put another word that was probably in the guided lesson. They're very squished or squashed. Yeah, several of us are mentioning in the chat that the word squamous, or sometimes you'll hear it said squamous, same thing, pick whichever way you like better. These are our flat, squished cells. Flat, squished cells. So when we're looking at them, because we'll start looking at them together, a lot of times they're going to look like this. They'll have a dark spot in the middle that is their nucleus. So my flat, squished cells, those are called squamous cells or squamous cells. When I talk about a cuboidal cell, not a trick question, what are cuboidal cells going to look like? Cuboidal cells, yeah, they look like cubes, absolutely. So sketches are going to make them look really like a cube, like a square. When we start looking at them on slides, they're probably not going to be quite like a square per se, but they're going to be just as wide as they are tall. So they're going to look like a wide circle. They're going to have a really big nucleus in the middle, a really big dark spot in the middle. So cuboidal cells are kind of cube-shaped, or they've got a really dark nucleus spot in the middle. They are about as wide as they are tall. When I talk about a columnar cell, these are cells that look like a column. That's how they got their name. They're columnar. And if you're describing what a columnar cell looks like, it's much taller than it is wide. It's kind of column shaped. So here's the big idea from this first activity we did with our classmates. When I talk about epithelial tissue, so you'll see the word epithelium after it, or epithelial tissue after it, its name tells me what's going on with its shape. Is it simple, one layer? Is it stratified, more than one layer? Is it pseudo-stratified? Is it faking it till it makes it, pretending it has more than one layer? Hey, I'll mention there's one other type of tissue that its name tells us something about it. That type of tissue is called transitional epithelium. Can you help me out in the chat? When we talk about transitional epithelium, what's the deal with the cells in this kind of epithelial tissue? Has anyone read about this? What's their deal? Transitional. Yeah, Ariel is totally right. Transitional epithelium, the shape of these cells changes. It transitions. So sometimes they look kind of cuboidal, sometimes they look kind of squamous. The shape is changing. So even the name transitional tells me about the shape. And in this particular instance, it's telling me that the shape changes. So keep that in mind with epithelial tissue. Their name tells us what they look like, or if you're looking at a picture of epithelial tissue, their appearance can help you predict what their name is. So we did some of that with our sketches over here. I'm gonna have us go nuts here. So my friends who were in group 1, I want you to match up these first 2 pictures here. Use that pencil at the top to match up the names of these first 2. My friends in group 2, I want you to go nuts here, and I want you to match up my second 2. So group 2 friends, let's draw a line to connect these 2 to their name. Group 1 friends, let's draw a line to connect these 2 to their name. Let's see how fast we can do it. So Kelvin, we're not going to talk a ton about glandular epithelium. You do want to know that that stratified cuboidal epithelium is found in glands, but we're not going to talk specifically. There's some special words about glandular epithelium that we're really not gonna talk about. Endocrine versus exocrine. That's going to be outside of the scope of our class. To the question about glandular epithelium. All right, so group 2 had fun drawing. They even gave us a little smile here. That's awesome. Remind me to give you guys your penguin for the day, right? Got to draw your penguin eventually. Okay, so start here at the top. We are looking at a type of tissue. Notice that I have 2 rows of cells here. 2 rows means that I have more than one. So this type of tissue is a stratified type of tissue. We had 2 options for stratified tissues. One of them is columnar, those column-shaped cells. One of them is squamous, those flat cells. These cells are definitely columnar, column-shaped. So this is stratified columnar epithelium. Hey, let me give you an important note. I want you guys to write this down because I got this email question a couple of times. When you're looking for the shape of stratified epithelium, when you're looking for the shape of stratified epithelium cells, we have to look at it's called the apical or the top surface. We've always got to look at the top layer. This is going to be really important when we start talking about things like the skin, where there's tons and tons of layers of cells. We always have to look at that apical surface, the very top surface. See, this one's really nice and cute. The layers of cells, they look the same. Same thing for the most part with this stratified epithelium here as well. But there was one on the homework assignment this last week where the bottom couple of rows of cells looked cuboidal, but that apical row, the top row, was columnar shaped. We don't care what's going on down at the bottom when we're naming our epithelial tissue. We only care about what's going on at the top to help us name it. So keep in mind, anytime that you have more than one row of cells, look at that very top layer. It's called the apical layer or the top surface. Nicole asked why that wasn't transitional. Because transitional the cells never look columnar. The cells are going to either be cuboidal or squamous. So transitional epithelium is going to look different. We never have really tall, skinny cells like this. But that's a good question. Another question about transitional epithelium: how many layers does transitional have? It varies, but it always is stratified. So we're asking a lot about transitional. Here's my hint for you in identifying transitional epithelium. Transitional epithelium. One of my colleagues says that kind of looks like clouds. So if you look at the apical layer of transitional epithelium, it's going to be really puffy because those are the cells out there when they're not stretched out. They look really wide, like wide cuboidal cells. So transitional epithelium is, and we're going to look at some pictures today, it looks puffy on the apical surface when it's relaxed. It's also not going to have, notice how these cells here have got one distinct line where the top of the cells are, or one distinct line where the top of the cells are here. When we start talking about transitional epithelium, that cloud appearance, we're going to see cells that are kind of all over the place. They're bunching all over. So that's what I mean when I say it looks kind of like clouds. So I promise we will look at transitional epithelium, but transitional epithelium has this bumpy appearance on its outer surface because those are the cells that sometimes look kind of cuboidal, and if your bladder is really full, they're going to be spread out and look pretty squamous. Okay, so we've got stratified columnar, column-shaped cells. We've got stratified squamous. Notice that these cells here on the top are really flat. Sometimes the shape of squamous cells, when you're looking at them, they kind of look like fried eggs. So what I mean by that is I've got my yolk in the middle of the cell, and the rest of the cell is around it. So fried eggs, squamous cells, stratified more than one layer. We've got one layer of cells right here. Notice that these cells are just as wide as they are tall, which means that they're a cuboidal cell, and there's one layer of them. So we identified that simple cuboidal. And then our last example down here is that fake it till we make it type of tissue. So remember, that's pseudo stratified. Here's a note that I probably should have said earlier too. When you're trying to figure out if we're simple epithelium or we're stratified epithelium, you want to look at the position of the nuclei. So notice here with a simple epithelium, I've got one row. All my nuclei are pretty close to each other in my row. When I look here at my stratified epithelium, I've got one row right here and one row right above it and another row. I've got multiple lines of nuclei. There are several nuclei in each row. When you look at pseudostratified epithelium, see how these nuclei are all over the place? They can't figure out if they're in a line at the bottom. They can't figure out if they're in a line at the top or in the middle. They're all over the place. So when we talk about pseudostratified columnar epithelium, the best way to determine it's pseudostratified is you're going to see these zigzagging nuclei all over the place. So pseudostratified, columnar because they're tall and skinny. By the way, let's talk about these little guys up here on the top of my pseudostratified columnar epithelium. These guys up here at the top look kind of like eyelashes. Eyelashes. Does anyone remember from working on your lab activity what the ones are that look like eyelashes? What are those ones called? Yeah, so these ones up here that I just completely drew on, these ones up here, we would call them cilia. So cilia are eyelashes. Well, they're not actually eyelashes. They look a lot like eyelashes though. They're extensions on the top of the cells. The goal of cilia is to move things. In particular, it's mostly going to be things like mucus or dirt or microbes. Cilia always move things like that. So when we talk about pseudostratified columnar epithelium, the full name you're actually going to see for this when you're looking at it in the packet, it's called ciliated pseudostratified columnar epithelium. That's going to be its whole name. So the whole name is also going to include this extra word here, ciliated meaning I have cilia on the top. Hey, yeah, I know, Jacqueline's like, goodness, long name, right? Hey, here's the good news. Back in the day, about 2 years ago, if you took this class, you would have to write that entire name from memory. The good news is we do not have to write that entire name from memory anymore. So we're happy that we don't have to do that, right? That makes a big difference. So we don't have to write the whole name anymore. But what we do need to know is this word ciliated means that we have cilia. You will also see some tissues that are called non-ciliated. Non-ciliated. We're only going to have non-ciliated simple columnar. Non-ciliated doesn't come in pseudostratified. This non-ciliated word here at the beginning means that we don't have cilia. That's all it means, don't have cilia. What we have instead is something that someone had mentioned in the chat. There's these things called microvilli. Microvilli, ooh, somebody wanted to color. Microvilli are places where I fold the plasma membrane of my cell to increase its surface area. Microvilli help if we're doing absorption. So if you are in the GI tract, my favorite system, that's going to help you to absorb nutrients or to absorb water. So if you ever see this terminology non-ciliated, it's kind of like microvilli are kind of like the younger brother of cilia, where everyone just always relates them to cilia. So I don't say that these tissue have microvilli. I say they don't have cilia. So non-ciliated simple columnar epithelium, these have microvilli. We're going to look at some pictures of these. Instead of eyelashes, microvilli kind of looks like peach fuzz on the top of these cells. So two different things we might find on the top of the cells. If we find eyelashes, we call them cilia. If we find something like peach fuzz, we call that microvilli. This slide looks real special, right? We wrote on it together, so it's perfect. All right, let's do a little matching. Full disclosure, I forgot to completely edit this slide. One of my types of tissue that's listed over here does not have a match for its location, and one of my locations doesn't have a match for its tissue. So I saw both of my groups, it looks like, highlighted this for me. We don't have the matching location for this one, and or the match of the name, and we don't have the matching location for this one, right? So let's start with the ones where Dr. Allis made a typo and didn't quite edit her thing. When we're talking up here at the top with the alveoli of the lungs, this is the location of which kind of epithelial tissue? Did we figure this one out? Who lives in the alveoli or the air sacs of the lungs? Yeah, so it's lots of typing, right? So we could abbreviate here. I'll help us for future reference. If we want to abbreviate, you can totally do this, right? I'll let you type it like that. The full name, like we're typing in the chat, simple squamous epithelium. So let me mention something here really fast when we're talking about locations, and in a moment when we're talking about functions. Hopefully you had a chance to find the tissue location and function guide that is posted in Visible Body or in the student Google Drive. When you look at that sheet, there are some locations that are highlighted bold, and there are some locations that are not highlighted in bold. The ones that you're responsible for knowing for homework assignments and for the exam are the ones that are in bold. So for each of these types of tissues or these locations that you see right here, these are the ones for each of my types of tissue that are in bold. You'll notice for the alveoli of the lungs, if you're looking at simple squamous epithelium on that location and function guide, there are other locations listed, but they're not in bold. Oh, perfect. Ariel said the link's in the guided lesson too. Awesome. Good. So it's all over the place. You'll notice that there are other locations of simple squamous epithelium that are not in bold. If they're not in bold, your teacher is 100% telling you to ignore them. You only have so much space in your mind to learn locations and functions. If it's something that's not in bold on that sheet, do not learn it. So you've got that tissue location and function guide. You've got your worksheet where you're trying to draw pictures. And if they're not pretty, I heard a couple of groups saying they're just not. That's okay. It doesn't have to be pretty. But you've got that space underneath those sketches that you're making that says the location and the function. What I want you to do is to write in that space only the ones that are in bold. Only learn what you have to learn because that's all there's space for. So the main location of simple squamous epithelium that we want to make sure we know, the air sacs of the lungs, the alveoli of the lungs. The other one that's on that sheet, we don't have it in bold anymore, but there's one right underneath it that's called kidney glomeruli. That's the blood vessels that live in the kidney. But hey, it's not in bold, so don't. We want to learn that simple squamous epithelium is in the air sacs of the lungs. Full disclosure, I forgot to trade this out with the name simple squamous. Stratified columnar epithelium is one of them that we don't actually have pictures of, which means that it's going to be hard for me to ask you questions about its appearance on homework assignments and on exams. So here's a wink, wink, nudge, nudge. Make sure we know its location and its function. I can't really ask you about it with a picture because we don't have a slide in our lab of this type of tissue. Can anyone help me out in the chat? Where did we figure out that stratified columnar was actually found? Yeah, so one of the best places for stratified columnar is those salivary glands. Salivary glands. I don't really have a slide that's just salivary gland tissue, so I can't really ask you where I find it. So salivary glands would be the location of stratified columnar epithelium. And if you have your tissue location and function guide, you'll notice that is their location is in the salivary glands. And when we look over at their function is secretion. And here's a note for us. Let me add that here. Function is secretion. When in doubt, if you're having a hard time figuring out the job of a type of columnar epithelium or a type of cuboidal epithelium, when in doubt, guess that those ones do secretion. Jacqueline's asking in class, what specifically does secretion mean? Secretion means that we build something and spit it out. So in the case of the stratified columnar epithelium that are found in salivary glands, this is the type of cell that makes saliva, that makes spit. When we're talking about the cuboidal epithelium that we find, for example, in the ovaries or in some of our ducts of glands, they're spitting out things like hormones or spitting out things like sweat. So secretion is just kind of a fancy word for I spit something out. So in the case of stratified columnar epithelium, they spit out saliva. In the case of glands, they secrete or spit out sweat for us. So I apologize for our confusion here. These guys don't match up, but here's what they would match up with. When we talked about the epidermis, which is also called, or if you were to put it in easy person words, it's the superficial layer of the skin. Can we draw a line to which type of tissue the epidermis matched up with? Which kind of tissue does the epidermis have? Yeah, somebody got some lines for me. The epidermis has keratinized, stratified, squamous epithelium. Again, remember that about a year ago, 2 years ago, you would have to write all of this. So we're really happy we're not writing all of this. Keratinized, stratified, squamous epithelium. Let's talk about this location really fast. This word superficial, can you help me out in the chat? What did superficial mean? That was a directional term from week 1, way back in the day when we were still sleeping, right? Yeah, so superficial means that we're on the outside. Specifically, the epidermis is the outermost layer of the skin, or it's the layer of the skin that you see when you're looking in the mirror. So the epidermis, this beginning part, epi, means above. We're going to talk about the epidermis next week because we're talking about the skin. Epidermis is the superficial layer or the outermost layer of the skin. That is made of keratinized stratified squamous epithelium. This keratinized word means that this type of tissue has keratin in it. Keratin makes things really tough or really strong. Keratinized stratified squamous epithelium. I've got lots of layers of cells that's stratified. Those cells are flat, that's squamous, and they're keratinized, meaning they're full of keratin, which is making them really tough or really strong. Keratinized stratified squamous epithelium, found in the outermost layer of the skin. You will notice on your location and function guide that there is also non-keratinized stratified squamous epithelium. This kind of epithelial tissue is not found in the epidermis layer of the skin. We would say that this kind of epithelium is found in places like the lining of the mouth or found inside the esophagus. I'm gonna run out of space. Okay. Non-keratinized means there's no keratin, but you'll want to have multiple layers. In places like your mouth or your esophagus where you're chewing food and then swallowing it, we want to make sure we have lots of cells there to protect us, but we don't want those cells to be really hard. So non-keratinized stratified squamous epithelium, the cells are not nearly as hard as the keratinized ones are. So we find them in different places because they have different anatomies, different structures. They do different jobs for us. The question in the chat is if these keratinized stratified squamous epithelial cells make calluses. Yes, that's one of the things that they make, but they're also found everywhere. So your entire outer layer of your skin is made of these keratinized cells. But calluses would have a lot of these cells there. Absolutely, there'd be a group of them. All right, we've matched up keratinized stratified squamous epithelium. Now we need to match up non-ciliated simple columnar epithelium. Where did I find non-ciliated simple columnar epithelium? There we go. Yep, we're getting a little line. GI tract, my favorite system. Remember, we talked about this on the previous slide. If I say that I'm non-ciliated, non-ciliated means that I have something called microvilli. Microvilli. And microvilli are really good at helping with absorption of nutrients. That's what they help me to do. So I want to have a type of tissue that's got structures. It's non-ciliated, meaning it's got microvilli. These structures are going to help me absorb nutrients. If I'm absorbing nutrients, obviously I'm in the GI tract. That's where the nutrients are, in the GI tract. So non-ciliated simple columnar epithelium. Function is helping to absorb nutrients. I do that in the GI tract. So by process of elimination, the last one we got here is that transitional epithelium. Transitional epithelium is the type that we said on the previous slide changes shape. So I find transitional epithelium in two of my organs of the body that change their shape depending on how full they are, and that is the bladder and the ureters. Ureters are the little tubes that connect to the bladder. They go from the kidney down to the bladder. Depending on how recently you use the bathroom, the ureters will expand or contract. Same deal with the urinary bladder. So in the very middle of your bladder, we have this type of tissue called transitional epithelium. Transitional epithelium, no, it is not found in the stomach. We actually have a different type of tissue there. The only place you see transitional epithelium is in the urinary bladder and the ureters. So to answer your question, Nicole, no, they're not found in the stomach. Urinary bladder and ureters. Okay, I told you guys that this is stuff for flashcards. A lot of stuff in our class is not stuff for flashcards. The locations of the types of tissues is stuff for flashcards. If I were making my flashcards, number one, I would make them today. Because if I don't make them today, I don't have enough time to study them. It's not worth it. So make your flashcards ASAP so you have time to study them. If I were making number two, here's how I would make them. On the front side of the card, I would say location of keratinized stratified squamous epithelium. That's the front side of my card. Back side of my card, the epidermis, and in parentheses, the superficial layer of the skin. That's one note card. The other card would say function of keratinized stratified squamous epithelium. And on the back side of that card, I would list my function like you see it on the guide. So protection against abrasion, water loss, radiation, etc. I would totally recommend splitting up locations and functions, because some of these types of tissues, it's going to be easier for you to learn its location than it is for you to learn its function, or vice versa. Maybe you'll be really great at the function and have a harder time with the location. So split it up, give each kind of tissue its own note card for its location and its own note card for its function. And here's the general rule with notes too. When you build them and you put them in a stack, as you go through and review those note cards in your stack, if you have correctly answered that note card like 2 or 3 times, that note card needs to come out of your stack. See, we have this tendency when we're using note cards to study to keep around those ones that we know the answer to right away because we feel really good. That's like, oh yeah, I totally know that keratinized stratified squamous epithelium is in the epidermis. Look at me, I got that one right. Okay, you got that one right, but maybe we got stratified columnar wrong, and maybe we got transitional epithelium wrong. We need to keep studying those ones more and not waste our time on studying the ones that we know. So when you're building your note cards, if there are a couple of type of tissues that you already know the location of, don't even build a flashcard for that one because you know that location. Time is precious, right? Especially this week when we have a ton of new information. Don't build flashcards for stuff that you already know. I know it feels sad because believe me, I love to make flashcards of information I already know, but I'm wasting my time. I'm patting myself on my back, and then there's 5 other things that I'm not learning. So use your time wisely. Build those flashcards. 1 card for location of a type of tissue, 1 card for a function of the type of tissue. And again, when you start getting it right, when you know that information, pull that card out so you know what you actually need to focus on. So there's my little flashcard tip for you guys. Hopefully that helps us save a little bit and focus where we need to. Really fast, matching up functions. With the functions, we've switched over from epithelial tissue to connective tissue. So when you are looking at your tissue location and function guide, not only are we learning those locations and functions of epithelial tissues this week, we're also doing connective tissues. So your homework assignment will help you to practice that. I'm looking at my guide really fast here. Looks like those connective tissue functions go all the way to the middle of page 3. So make sure we're learning those functions as well. Really fast, let me just put it to the class so we don't spend too much time here. Can we draw some lines? It's a free-for-all. Anyone can match up anything. So groups 1 and 2, let's have at it. Let's match up our types of tissue with their function. Looks like I had 1 friend who connected the lines for us. That's okay. My 1 friend is right. So let's talk briefly about what we see here. First thing I want you to notice, at the top, my 2 types of connective tissue that are here at the beginning, dense irregular connective tissue, dense regular connective tissue. These types of connective tissue, we can learn something about their appearance based on their name. This word dense at the beginning of both of these means that these 2 types of tissue have a lot of proteins in them. Connective tissues have a lot of, we used this word last week, we called it extracellular matrix. Extracellular matrix. Remind me, because we've talked about this a lot, extracellular. Where's that found? Extracellular. Yeah, a couple of us have chimed in. Extracellular is outside the cell, on the outside. One of the big things we find outside the cells in connective tissue are proteins. That's one of the big ones. So dense, irregular connective tissue has a whole bunch of proteins outside the cells. Those proteins are arranged irregularly, meaning they're going all over the place. It's a big mess. If I've got proteins that are going all over the place in all kinds of directions, that's going to help me be strong in multiple directions. I've got proteins that go up and down. I've got proteins that go sideways. I got proteins that go in an angle. By having proteins going in multiple directions, I'm going to be strong in multiple directions. So dense, irregular connective tissue, because those proteins are arranged irregularly, I've got strength in all kinds of directions. Dense, regular connective tissue, all of those proteins are arranged regularly. They're all arranged in one line. I've got a whole lot of proteins going one direction in one line, which means I'm really strong in that one direction. So dense, regular connective tissue found in things like tendons or ligaments that pull, that are attaching things there, withstanding that pulling in one direction. So your arm going up and down, for example, that's what dense regular connective tissue helps with. Dense irregular connective tissue is found in places like the dermis, which is deep, to use our directional term, deep to the epidermis, space. Dense irregular connective tissue is found in that layer underneath the outside of your skin, so that when you move your fingers, when you move your toes, when we're pulling in a whole bunch of different directions, your skin doesn't rip, for example. So dense irregular connective tissue, proteins go in all kinds of directions. Dense regular, they go in one direction. So the question that I see in the chat is, are these proteins in interstitial? So I think what you're getting at, we talked about the fluids that are found in different places in the body. So when we talk about extracellular matrix, that would include, if we were to make kind of an equation for it, that would include interstitial fluid, and then it would also include, not in the fluid itself, but around it, those things like proteins. And we're also going to see things like minerals, for example, calcium or chondroitin sulfate. So it's found in, we could say it's found in the interstitial space, maybe, because interstitial means between cells, but it's not part of the fluid. These proteins are not floating around in the fluid. So I'm not sure if that quite answers your question. It's different than interstitial fluid, but it would be in the interstitial space. Yeah. Okay, let's talk about reticular here really fast, because that ties into proteins as well. Reticular connective tissue has what are called reticular proteins in it. Reticular proteins, that's how it got its name. Reticular proteins look a lot like a net, as in the kind of thing, if you were to go fishing, you would have to help you catch things, a fishing net. Reticular proteins branch all over the place, and like I said, they kind of form a net inside our tissues. So reticular connective tissue is used to help us filter our blood, so to pull out blood cells that are broken and not functioning anymore, to pull out bacteria or viruses. We're going to see reticular connective tissue. Really the biggest place that we see reticular connective tissue is in the lymph nodes, which are part of your immune system. So catching those bacteria, catching those viruses. We also see reticular connective tissue in places like the spleen, where we're doing a lot of filtering of our blood. So reticular connective tissue found in places where we pull out bacteria, found in places where we pull out blood cells to recycle them. Reticular connective tissue with these branching proteins. Our last kind of tissue is the last group of tissues, and that's cartilage tissue. Now, let me give you a hint here. This is going to become more important when we start being in lecture number 2, but you're going to see a word called chondrocytes. At the end of the lesson number 4 lab packet, it asks you about types of cells. One of the types of cells are called chondrocytes. This beginning part, chondro, right here, chondro means cartilage. So one of the types of cartilage tissue we have is called fibrocartilage. We also have what's called hyaline cartilage or elastic cartilage. Anytime I have cartilage tissue, the cells inside of it are called chondrocytes. So chondro means cartilage. Chondrocytes, cartilage cells. When I talk about fibrocartilage, not only do I have these little chondrocyte cells, I also have a whole lot of fibers. So fibrocartilage has a whole lot of fibers inside of it. Fibrocartilage is really good at holding things together because it has these strong fibers. It's also good at absorbing shock. Now, when we think about absorbing shock, it helps us to understand what that means to think about where fibrocartilage is found. It's actually found between the vertebrae of your spine. So one of the locations you'll see listed, it's called the intervertebral discs, vertebral discs. So between the vertebrae of the spine. You also see it in a place called the pubic symphysis. Pubic symphysis, that's between the halves of the pelvis. Remember, we had the pubic bones in the front. Holding them together is something called the pubic symphysis. And so that's a group of really strong cartilage, fibrocartilage, with protein fibers and cartilage connective tissue. So cartilage tissue with lots of fibers in it, that's good at holding your pelvis together. That's also good at absorbing the shock that comes from you walking or that comes from your head resting on the top of your spine. So I point out the locations of these types of tissue because it helps you to get an idea of what their functions are as well. Just like we said that anatomy and physiology are related to each other. So let's remind ourselves here, anatomy, what's there, and physiology, what it does. Just like these two things are related to each other, functions and locations are related to each other. So make sure we can learn both the functions and the locations on each types of tissues. I know it's not sexy, but the best way to learn these types of information, the functions and the locations, it's just practice. We've just got to be seeing it over and over and over again. So that's why I told you guys, make your flashcards today or make them tomorrow. We just got to start practicing the locations and the functions. And I'll mention this, you guys have your assignment that's due on Sunday. I really want you to try to know as many of the locations and functions from memory as possible by Sunday. Because the thing is, our lab exam, I know we don't want to think about this, right? Does anyone happen to know when is the lab exam? Do we know how long it is till we start taking that lab exam? Yeah, so it opens up October 6th. I believe. Let me check my list to make sure I don't tell you wrong here. Yeah, so it opens up October 5th and it has to be done by October 6th. Pulling out my handy dandy calendar here to say that that is 3 weeks from today. 3 weeks from today that exam opens up. So we cannot memorize all the locations and all the functions in one night before that exam. We can't put this off for 2.5 weeks till we actually start to memorize these things. We have to do that now. So please start with your flashcards now. please start memorizing now. It's coming up fast. That exam is going to sneak up on us. After we finish the lecture exam, then we really got to start working toward that lab exam. So these locations and functions, flashcards now to start memorizing them now. I know I see the emoji that Jacqueline's put in the chat. We haven't had an emoji party today. Show me how we're feeling. Jacqueline's feeling stressed, and she's sweating bullets here. Ariel's screaming. Yep. We're feeling it 100%. So I know that this is, yeah, they're all related to each other. The emojis we found are all related. I know this is a big week. I know that this is stressful. I'll remind you guys that you have that group me chat with your classmates. 100% when you're up late tonight or in the next few days, you're up super late studying these locations and functions, shoot out a quick text into the void and say, hey, is anyone else up studying these things too? I bet that there will be classmates that are doing the same thing. Or, you know, if you shoot out into the void, oh my gosh, I'm so stressed about these functions and locations, I bet you're going to get a whole lot of chatter that says, me too. This is not easy stuff. So please know that your classmates are all in the same boat too. Again, it's not sexy, but the way that you got to learn these functions and locations, it's just repeated practice. So start doing that now. Maybe start quizzing each other in GroupMe. Whatever you can do to make it fun, to make it the monotony of it go away. It is monotonous, but that's what we got to do to learn these locations and functions. Before I start looking at pictures of tissue types with you guys, before we do a crash course through some of these tissues, do you have any quick questions for me related to some of the stuff we've talked about? I already got our emojis, so I know how we're feeling. I'm going to get my screen ready for us here. Okay, well, I haven't had anyone chime in any questions, so I'm going to start working on pictures with you. Okay, I did get one question. Good question. The question said that on the location and function guide, it will say reticular loose connective tissue, or you'll see things like dense elastic tissue. Those words are important. Well, okay, so there's two types of tissue. Let's talk about what that word means. Two types of connective tissues in the body. There's dense connective tissues that have a whole lot of stuff packed close together, and there's loose connective tissues where there's space in between those types of tissue. If we were doing the fill in the blank type exam where you had to write the entire name of the types of tissues, you would 100% need to make sure you memorized whether it was a loose or a dense tissue. Since you are matching up names that are given to you, you don't need to focus too much on the loose versus dense. But as you're studying, use that as a tip for yourself. If I'm looking for dense, elastic connective tissue, that tells me I'm going to see a lot of stuff squeezed in that type of tissue. When I'm looking at reticular loose connective tissue, my stuff is going to be more loosely arranged. So use it to help you understand and to help you study. I am not a stickler about dense versus loose, which is why you'll see me just call it reticular connective tissue. Technically, yes, reticular loose connective tissue, because there's space in between the cells. Ariel mentioned that we missed spongy bone and compact bone on the guided lesson. Interesting. I will look into that. I'll make sure that I show you guys that kind of tissue. That was just an omission. Somehow that's missing. So I'll work on editing that, getting that added. All right, crash course. And here's what I'll say. I have to scoot out at 11 because I'm sharing my camera. But today, this evening, we can spend a little more time on histology. So we'll do a little bit of lecture stuff, but we'll also do some lab stuff tonight. So if we don't get to your question that you have with tissues, plan to come back tonight at 5 if you can. We will do some more lab stuff. No, Karina's like, no, you can't go. Right? I would stay here forever if I could to help us out. But alas, I have to today. So come back tonight if you can. We'll do histology more tonight because I know it's important. All right, first type of tissue that we're looking at right here, and I apologize, I can't. So I tried to upload this into Blackboard where we could write on it, and it told me, oh, your file is too big. So I can't write. So maybe you guys can write in the chat to help each other out. What we're looking at right here, this is simple squamous epithelium. Actually, I can write in the chat. Here we go. Simple squamous epithelium. Simple squamous means there's one layer of cells, and those layer of cells are flat. Those cells are flat. This line that I see right here is a layer of cells. And here's a line with a layer of cells and a line right here. So my image over here on the right is one appearance of simple squamous epithelium. The other appearance that I see over here, it's still simple squamous epithelium, but it just looks a little bit more wiggly back and forth or weepy almost. I don't know, whatever word you want to use for it. With simple squamous epithelium, we did the matching in our groups where simple squamous epithelium is found in the air sacs of the lungs. So these big open spaces that you see right here on these slides, literally those are open spaces in your body. These are places where the air would be found in your body. So when you look at a type of tissue that has a line of cells surrounding a big open space in the middle, this is simple squamous epithelium. And we're looking at a slide of the lungs. Simple squamous epithelium. Next type of tissue that we're looking at right here, this is simple cuboidal. Oops, spelled that one wrong. Cuboidal epithelium. Simple cuboidal epithelium. Simple still means that there is one layer of cells. Cuboidal means they're about as wide as they are tall. Now let's talk about how this is one layer of cells. Simple cuboidal epithelium, see this white space right here, and this white space right here, and this white space right here. We're looking at, in this picture, a cross section of the kidneys. The kidneys have a whole bunch of tubes in them that you use to filter out your blood to make urine. So each of these white spaces that I see right here is actually an open space inside your kidney. The cells that are found around it in a circle, and you can kind of see the dark line on the outside of this circle, this is the epithelial cells. So this is one layer of cells that are arranged around this opening, the tubule of the kidney. This one layer of cells is simple cuboidal epithelium. And here's another group of cells, how they kind of have a dark line on the outside of them. That's simple cuboidal epithelium as well, around their open space in the middle. Bounce over to this one here. I've got an open space and this row of cells around it. This is another example. Here's another row of cells with their open space in the middle. Both of these tissue images that I'm looking at show us simple cuboidal epithelium. The ways that you're going to recognize it, you're going to see these circles of cells. They're arranged kind of in groups, and they've got that white open space in the middle. So the white open space in the middle and that layer of cells, that's a bad one, right? Let's go over here. This layer of cells that I can see. I told you before, remember that cuboidal cells, they might not look actually like cubes, but the big thing with those cuboidal cells is that the nucleus is so big. So the nucleus inside these cells looks really big. It's really wide. Let's compare that. Let me go back, if it'll let me. Back to these cells. The dark spot that you see here, that's the nucleus in each of these cells. That nucleus is squished. This whole cell is very flat. When we're looking at those cuboidal cells, the cells are still going to maybe look a little bit circular, but that nucleus is really wide. I can see it in the middle. Simple cuboidal epithelium. These are the kinds of images you'll see for that type of tissue, simple cuboidal epithelium. Let's look next at our two types of columnar epithelium. So these are column-shaped cells. Now, I think you guys can help me out in the chat. When we are looking at, let's start with this image over here. On the top of this image over here, see how I've got these eyelashes going on? What did we say it was called when a cell had eyelashes? Or what are these eyelashes up here on the top of these cells? All those eyelashes. Yeah, so these eyelashes that I see on top of this tissue, these eyelashes are cilia. Types of cells that have eyelashes are called ciliated, meaning they have those eyelashes. When I look over here at these cells, and by the way, these dark spots here, that's the nuclei of these tissues. So this tissue, I've got one layer of cells. It's kind of curving all over the place, but it's one layer of cells. Notice that these cells don't have all of the eyelashes that I see over here in my other image. These kind of have a peach fuzz appearance. See that little peach fuzz, the fuzzy stuff on the top? Actually, it's funny for me right now to talk about peach fuzz because I've got my 3-month-old and he totally just has peach fuzz. So here's his head here, just a little bit of stuff on the outside. That little bit of stuff on the outside were those things that we talked about called microvilli. I'll type them back in so we remember. Microvilli. That's all these little things that we see on the top here that kind of look like peach fuzz. When you have a type of tissue, a type of columnar tissue, there's my qualifier here. When I have columnar tissue that has peach fuzz on the top of it, I call it non-ciliated. Non-ciliated, meaning it doesn't have those eyelashes. We have one layer of cells and they're column shaped. So we're going to call it, here's its full name coming at you in the chat, non-ciliated simple columnar epithelium. No eyelashes, just peach fuzz up here. One layer of cells, because I got one line of my nuclei going around here. And these cells are very tall and skinny. This one over here, non-ciliated simple columnar. And we look over here, we already talked about how these ones are ciliated because they have the eyelashes on the top. These cells are tall and skinny as well. But check this out. Remember how I told you that sometimes we've got types of tissue that their nuclei are all over the place? See how I can see it going up and down everywhere inside these cells. These are my fake it till you make it cells. So let me give you the full name of the second tissue on here. Ciliated Pseudostratified Columnar Epithelium. Ciliated because I can see my little cilia on the top here. I can see my eyelashes. Columnar because they're tall and skinny. And pseudostratified because they're going nuts all over the place. So nuclei all over the place, that's pseudostratified. One line of nuclei, like I see right here, that's simple, excuse me. You'll notice, by the way, in your lab packet that we gave you a picture of what stratified columnar epithelium looks like. I don't have images in the slideshow to show you that because, again, I don't have any slides in the lab that show that, but make sure you can recognize it in that picture and really make sure we know where it's found and what it does, because I don't have a lot of good pictures of that type of tissue. So actually, I think it'll let me draw. Let's see if it'll let me draw. Stratified columnar, you're going to see rows of cells that are divided in the middle, nuclei at the bottom, nuclei in another row. So we gave you stratified columnar. Guess I can write a little bit. It's going to be hard. Stratified columnar. You've already got that one. Picture of that in your notes. But you do need to draw a picture for yourself of the simple columnar, the non-ciliated simple columnar, as well as the ciliated pseudostratified columnar epithelium. When I look at this slide here, can you help me out in the chat? What two types of tissue do we see here? Any guesses? By the way, you can totally abbreviate because these are long words, right? Oh, Jacqueline says she wants to cry. Don't worry, we're working on practicing here. Okay, so a couple of us have honed in the word ciliated. When we're looking at, is it on the left or the right, or both, that I have cilia on? Left picture, right picture, or both? Yeah, left picture has cilia. See all these little things that are sticking up right here? We've got cilia over here. We have our cells that I find inside here. We know that they're tall and skinny cells. Hey, by the way, here's the other study pro tip. Cilia only found on columnar epithelium. So these are some type of columnar epithelium. We are debating in the chat if it is simple or pseudostratified. When I look at this image, see how there's some nuclei here and there's some nuclei here. We're bouncing all over the place with these nuclei. Since I've got nuclei all over the place, this is my fake it till we make it. This is ciliated pseudostratified columnar epithelium. Nuclei bouncing all over the place, pseudostratified. Cilia at the top makes it ciliated. So this over here is ciliated pseudostratified columnar epithelium. Over here, notice how I've got one nice clean row of nuclei. One nice clean row of nuclei here means that this is simple epithelium. And because I don't see eyelashes, see these distinct little eyelashes? I don't have distinct eyelashes. I got this fuzzy stuff that I see here. Since I've got fuzzy stuff, I call it non-ciliated, simple columnar. Again, simple for one row. This fuzzy stuff tells me it's non-ciliated. Really fast. Okay, two things really fast, and then I got to scoot. We'll do connective tissue tonight. So come back tonight, we'll do connective tissue. This right here is our two appearances of transitional epithelium. First appearance of transitional epithelium, we have this yellow color type of tissue that you see here. Notice how at the top though, how it kind of looks like a cloud, how it's bouncing around the surface of it. So first image of transitional that we see here shows us that cloud appearance at the very top layer. Lots of layers of cells and they look all kinds of funky shapes, right? Second appearance of transitional epithelium, I've got this purple and pink slide, but again, notice how at the top it's kind of bumpy on those cells. So this bumpy appearance that we see up here where it's not really smooth like some of my others, that's transitional epithelium, kind of looks like a cloud. Last thing to mention really fast. This is my two types of stratified squamous epithelium. Remember that we said with stratified epithelium, you have to look at the apical surface, the very top surface. So both of these types of tissues, when I look at the top surface, they're very flat cells. This over here, I'll type into the chat, is called non-keratinized epithelium. Non-keratinized, put the whole name, stratified squamous, abbreviation. Non-keratinized, because it's not filled with keratin. These cells are all alive, they're still attached. Over here on the right, we are seeing keratinized stratified squamous. Keratinized because these cells are filled with keratin. I know they're filled with keratin because they're falling off, they're dying. So keratinized stratified squamous epithelium over here on the right. Non-keratinized stratified squamous epithelium here on the left. I know that we probably have so many questions. I am going to have to end the recording now. I'm going to stick around in the chat if you want to chat some questions at me. But again, tonight at 5 o'clock, 100%, we will probably spend our whole time doing doing tissue stuff. So bring questions if you want to talk about them together. Let's bring them tonight. If you have a simple question you want to try in the chat, go for it. I will get this posting or get this recording up for you as soon as YouTube processes it. So good luck with your working on this stuff. We'll continue lab tonight. Nicole, I don't know how much lecture time we're going to have tonight. I asked you to be through about page 6 or 7. So do as much as you can to get you ready. We'll see if we have time for lecture stuff tonight. So I'm going to stop my recording